Bismillah. Assalamu alaikum, this is Dr. Amira. Today we're going to talk about patterns of single gene inheritance, and this is the first part of the lecture, and we'll talk a bit more in the second part. Please refer to pages 109 to 124 in the textbook, Thompson and Thompson 9th edition, for more information about what is covered in this lecture. So most of this lecture is going to be about introducing some terminologies to you, some important terminologies and definitions. So let's start with what is a genotype. So a genotype of an individual at a particular locus is determined by the alleles in that particular locus. So what is a locus? Locus is each point here. You can see the uppercase A and the uppercase B. Each of that is a locus. And similarly, in the diagram, you can see the lowercase a and the lowercase b. Each of that is also a locus. And the genotype is a combination of what is present in both the mother's copy and the father's copy of that particular gene or DNA segment. So the uppercase a and the lowercase a in combination is one genotype, and the uppercase b and the lowercase b in combination forms another genotype. So for autosomal loci, so autosomal is the first 22 chromosomes, as well as the X-linked loci in females, the genotype includes the alleles in the two homologous chromosomes. So two homologous means these similar chromosomes. So if it's a chromosome number 1, it's a maternal chromosome and the paternal chromosome, and the allele that they have on the maternal copy and the paternal copy together forms a genotype. So what is an allele? Allele is one of the alternative versions of a gene or a DNA sequence at a particular locus. Okay. Now, more broadly speaking, not just in one individual, a genotype comprises of all of the alleles that could occur in that particular locus. So you know that there's A, T, G, and C, the nucleotide bases. So if you're talking about just one particular segment of the DNA, it could be A in one spot, and depending on, it could be T in the maternal allele and the paternal allele, or it could be different combinations of that. It could be AA, it could be AT, it could be AG. So it's really important to know that all of those combinations, as long as there is one in the maternal copy and another in the paternal copy, that together forms a genotype. So another terminology also important to understand is haplotype. So haplotype doesn't involve the two copies, not the maternal and the paternal copy, but it involves two or more neighboring loci on one of the two homologous chromosomes. So in this case, go back to the diagram here, you can see the uppercase A and the uppercase B are neighboring loci on one particular chromosome, and they will form one haplotype, whereas the lowercase a and lowercase b together will form another haplotype. So these are terminologies that are important to understand. Another important terminology is phenotype. What is a phenotype? A phenotype is how a genotype is expressed in the body. In other words, what the genotype does to the body. Phenotype is the expression of the genotype as a morphologic, clinical, cellular, or biochemical trait. So it doesn't necessarily need to be a disease. It could be a blood group, for instance, which we can't see from the outside. We'll only find out when it's actually measured. Okay. So it can be clinically observable or may only be detected by blood or tissue testing. A phenotype can be qualitative phenotype, which means it's either present or absent. So presence of disease or absence of disease. Or it could be a quantitative phenotype, so blood glucose levels. So in a quantitative phenotype, we know that blood glucose levels can be high, very high, normal, or low. So all of those are different quantities that we can measure. A few more terminologies. These are also important to understand. So wild type. Now, wild type doesn't necessarily mean that, animals in the wild have got a particular genotype. It's a funny terminology. However, it's important to understand what a wild type genotype is. So wild type is basically a non-disease genotype, but we've come a long way now, and we know that a lot of different types of genetic problems can occur, not necessarily genetic diseases, but there can be different traits. Now, some of you might have heard about Human Genome Project. So the Human Genome Project created a human reference genome. So the Human Genome Project was where a number of people, I think around 20, there was one person whose genome was mostly used, but there were 19 other people that they used segments of their DNA. And they used, this was completed, it took about 13 years, completed in 2003. It took them a long time to study each nucleotide base, so sequence each nucleotide base, and they tried to map out the human genome. It's not a perfect genome. There are still lots of gaps in it, but this is considered to be a reference genome, and anything within the reference genome is considered as the wild type. Lots of different iterations or different versions of the human reference genome have occurred ever since the first human genome project was done. So now we know that it's an artificially assembled genome, and it's considered to be free of disease, generally speaking. It's not the most perfect genome, but that is considered as the human reference genome, or the wild type, against which we look for any differences, and anything that is different from that is called as a variant. So I've included a URL here under the Human Genome Project, so if any of you is interested, you can go into that URL and have a look at a fact sheet about the Human Genome Project, which is quite interesting. So a variant is when the allele differs from the human reference genome. A pathogenic variant is a variant that causes disease. Another definition that's also important to know is pleiotropy, which means that there could be different phenotypic manifestations in different organs from the same disease-causing variant. For instance, let's take an example of a gene called BRCA, also known as BRCA. So BRCA1 is a gene, so BRCA1. It causes breast cancer in some females, but it can cause ovarian cancer in some other females. So even within the same family, with the same type of gene variant, there could be pleiotropy. So there could be some females within the family who have breast cancer, and some females can have it at an earlier age. Some other females have it at a later age. Some females may not develop breast cancer. They might actually develop ovarian cancer. So there's pleiotropy, even though the same disease-causing variant is inherited in various family members. Okay. The next thing also to know is Mendelian type of inheritance. So Mendelian inheritance is a single gene inheritance type pattern, so autosomal recessive, autosomal dominant, X-linked, all of these are single gene disorders, and these are the types of inheritance patterns that were classically described by Gregor Mendel. Some other things that are important to know is penetrance and expressivity. Penetrance is the probability that an allele will have any phenotypic expression. So complete penetrance means everyone with the disease, or everyone with the particular gene problem, will have the manifestation of the gene problem. Okay? Incomplete penetrance means even though a person might have inherited the genetic problem, they won't necessarily have a phenotypic manifestation. So they'll silently carry that, but they won't actually have any phenotype. So if you refer to the diagram at the top, you can see incomplete penetrance, where all of these people have the same gene problem, but only a few of them have developed the actual disease. They're showing manifestations of the disease or the phenotype, whereas the others have not shown the manifestation of this phenotype. So this is an example of incomplete penetrance. Expressivity is different to penetrance. It means that the disease can be variably expressed. So it could be that one person has more severe disease, another person has less severe disease. Or it could mean that one person develops disease at a younger age, and another person develops disease at a slightly later age, even though they have the same genetic variant. So that's something that's described in the diagram here, where you can see variable expressivity. So one person, the one in the darkest shade, is more severely affected, and then there are lighter shades who are not as severely affected, even though they all carry the same genetic variant. So penetrance, it's either yes or no. Expressivity is difference in the severity of the disease, and also there could be different phenotypic manifestations, like what we talked about in the BRCA1 example. So even though they carry the same variant, there could be variable expressivity in the family members, with some developing breast cancer, some developing ovarian cancer. Now, this is a standard pedigree and the standard pedigree symbols and terminologies. Pedigree is a formal, standardized way of drawing a family tree. So many people have different ways of drawing a family tree, and it gets confusing when you're trying to discuss this information with other medical professionals. So there is now some standardization, standard symbols that need to be used if you're drawing a pedigree for a patient who comes to you and you're taking a family history. So some important symbols, I'll let you go through all of these at your own time, but I'll just highlight some important symbols. So squares are males, circles are females, and if it is shaded inside, it means that they're affected. And if there's multiple shading, different segments, it shows that they've got different types of phenotype. So it may mean, for example, going back once again to BRCA1, if there are two types of shades, it means that, a person has breast as well as ovarian cancer, and you have to put that in the legend separately. If you put a diagonal line across, either a circle or a square, it means that that person is deceased. And you can see the symbols for twins, monozygotic twins, dizygotic twins as well here. And going to the third column, you can see consanguinity. That's another important symbol. So there are two lines. You join, if there is a relationship between a male and a female, if they're joined together, marriage or union, then there is a single line. But if they're consanguineous. Consanguineous means that they have a common ancestor, like cousins, first cousins, second cousins, or related within the family, blood relatives. You have two lines to show that they're consanguineous. I'll leave you to look at the other symbols by yourself, but it is important to know that there is standardization, and this is the only way we should be drawing pedigrees. Now, this is how a pedigree is drawn. You can see a circle here in the middle. In row 3, number 5, is the proband. The proband is the first person who manifests the disease and usually is marked by an arrow. So you can see here. And this particular picture just shows us the degree of separation of the other relatives from the proband. So you can see we don't normally put numbers and degree inside the square or inside the circle. But in this case, you can see we've drawn that. It's been drawn to show the degree of separation. So the siblings are first degree, so 1 degree removed from the proband. Similarly, the parents are 1 degree removed, and the children are also 1 degree removed. So they're all called first degree relatives. If they're 2 degrees removed, like grandparents on both sides, they are second degree relatives. And if they're further removed, like, say for instance, cousins, they're 3rd degree relatives, so on and so forth. So it's important to know the different degrees of your relation. And some common pedigree terminologies are kindred, which means extended family, which is depicted in the pedigree. Proband, also being called propositus or index case, is an affected individual through whom the family is first brought to medical attention. Consultant is the person who consults a medical professional. They may or may not be affected. Sibship are all the brothers and sisters in a family. Now let's go into the patterns of inheritance. So the first two here, autosomal and sex chromosome, these two are the Mendelian patterns of inheritance. And then we've got the non-Mendelian, which is mitochondrial. So you may have learned from your previous lectures that most of a DNA is within the nucleus, but there is also some DNA outside the nucleus in the mitochondria. So these are different types of inheritance. It's a mitochondrial inheritance, and we'll learn about that later. But we'll concentrate right now on Mendelian inheritance patterns. Autosomal means spanning chromosomes 1 to 22, and sex chromosome inheritance is X and Y chromosomes. It could be dominant or recessive. Note that there's only one copy of the Y chromosomes in chromosome in male, therefore it cannot be recessive. And we'll talk about the relevance and the difference when it comes to sex chromosome inheritance pattern in a little while. Let's start with autosomal recessive inheritance. So in autosomal recessive inheritance, disease-causing variants are present in both the inherited alleles, the maternal as well as the paternal. So there are some genes which are autosomal recessive, and you need to have a problem in both the maternal copy, so the copy that's inherited from the mother, as well as the paternal copy, the copy that is inherited from the father, for a person to actually develop the disease. An autosomal recessive disease can be homozygous when both the maternal variant, gene variant, as well as the paternal gene variant are identical. They're exactly the same. Compound heterozygous is when both genes are not working properly, but there's a different variant in the paternal copy and a different variant in the maternal copy. Okay, so homozygous is when it's identical. Compound heterozygous is still both copies are not working normally, but there are different genetic problems in the same gene, but from the mother it's a different problem, and the father it's a different problem, but it affects the same genes. Okay. Unaffected heterozygotes are people with the disease-causing variant in only one allele. So parents of a person with an autosomal recessive inheritance, if they're unaffected, are obligate carriers. So they will carry one copy which is not working, but they didn't have the disease themselves because they had another copy that was working. So these are carriers. And similarly, an affected person, if they have children with an unaffected person, so the other, if their partner does not have any gene problem, then their children will all be carriers once again. So if there are two carrier parents, the risk of transmission of the autosomal recessive disease to their offspring is 25%. In other words, it's a 1 in 4 chance. It's really important, if you've identified that it is an autosomal recessive condition, an autosomal recessive disease, to check for consanguinity. Consanguinity means that there is a common identifiable ancestor. Okay, so there could be first cousins, second cousins, or something like that. So you need to check to see if they've got a common ancestor, because they could have identical alleles inherited from both parents. And that increases the risk of autosomal recessive conditions. So you may have heard of Punnett tables before, or you may not have heard. A Punnett table is basically where you put the genotype of one parent at the top and the genotype of the other parent to the left, and then you map out the different combinations of genotypes that their offspring could have. So in this case, let's say that there are two carrier parents. So the lowercase r is where a pathogenic allele is present in an autosomal recessive condition. It's lowercase. It's usually shown in lowercase. So you can see here that there are two carrier parents. They didn't have the disease themselves, but when they have children, what will happen is that if it is 2 uppercase R, which is unaffected, or wild type allele, then the child will be unaffected. So it's a 1 in 4 chance, or a quarter chance of them being unaffected. Or they could be carriers by having 1 affected allele and 1 unaffected allele. So it's a 2 in 4, or a half a chance of them being carriers like the parents, or 1 in 4 chance of them being affected. So that is when there is a combination of 2 carrier parents. What if there is 1 carrier parent and 1 affected parent? Okay, so if there's 1 carrier parent to the left and 1 affected parent has 2 alleles that are both affected, so 2 lowercase r's there, then you can see that there is a 2 in 4 chance of the children being affected. But if there are 2 affected parents, all of their alleles are affected, therefore all their children will have 2 copies of affected alleles and therefore be affected themselves. So what are the characteristics of autosomal recessive inheritance? Phenotype is typically seen only in the sibship of the proband, not in the parents, offspring, or generally not in other relatives unless there is multi-consanguinity within the family. So brother or sister could also be affected, but the parents or the children are generally not affected because they're carriers, unaffected carriers. Males and females are equally likely to be affected. Parents of an affected child are generally asymptomatic carriers of pathogenic alleles. In other words, they're obligate carriers. It is important to look for consanguinity, especially likely if the gene responsible for the condition is rare in the general population but seems to be occurring a lot in a particular family, the disease. The recurrence risk for each sibling is 1 in 4. So if there is an affected child and if you're seeing the parents and if they ask you, do you think I'll be having another child? Then you tell them that there is a 25% chance. If you've identified that they're both carrier parents, you tell them that there is a 25% or a 1 in 4 chance for each of their pregnancy to also be affected. Okay. So unaffected siblings of the proband, even though they're not affected, what is the chance that they could be carrier? It's 2/3. It's not 2/4. It's 2/3 chance of being a carrier, okay, because they're not affected. So you take out the affected chance. Let's talk about autosomal dominant inheritance. Autosomal dominant inheritance is, there are certain genes where the disease-causing variant is only present in one of the alleles. So if just one allele is affected in some genes, which are autosomal dominant genes, that is sufficient or that is enough for the person to develop that genetic disorder. The phenotype usually appears in every generation. Each affected person will have an affected parent. There are some exceptions to this rule, so please pay attention. There could be new mutations, which means that the parents won't be affected and the child has developed a new mutation. This is called de novo. There could also be non-penetrance. So we talked about penetrance previously. So it could be that one of the parents is not affected because that gene is non-penetrant in them. The gene problem has not caused any phenotype, whereas a child has shown complete penetrance. Okay, or it could be there could be variable expressivity, which means that, say for instance, going back to the BRCA1 example, if we see a patient, she's got breast cancer, and we ask anybody else in your family who's got breast cancer, does your mother have breast cancer? And if she says, no, mom did not have breast cancer, then, you wonder if there is ovarian cancer in the family, because it could be variably expressed. So there could be ovarian cancer in the family. Then you work out that, oh, there might be an autosomal dominant gene, most likely BRCA1. And also, if it is inherited from the father's side, for instance, they're not going to have breast cancer. Males don't get breast cancer, generally speaking, with the BRCA1 gene problem. So then, it might look like nobody else has got a family history because only males have carried it. But males generally don't develop breast cancer with BRCA1, so it's variably expressed in males and females. So that's another thing to think about. So autosomal dominant genes may not look like it's inherited in each generation if there are new mutations, in other words, de novo, or if there is non-penetrance or incomplete penetrance, or if there is variable expressivity. There is a 50% risk for transmission to offspring, assuming that the other parent is unaffected. Unaffected family members do not transmit the phenotype to their children, and males and females are equally affected and are equally likely to transmit the phenotype. So here's a Punnett table for various autosomal dominant combinations. Let's say that there is one affected parent and one affected parent, and you can see here. So in autosomal dominant, uppercase is generally the affected genotype. So you can see here that there is a 1 in 4 chance. 2 in 4 chance for the child to be affected, so 50% chance, and 2 in 4 chance, or 50% chance of the child not being affected. So in autosomal dominant conditions, it's a 50% chance of either being affected or not being affected, depending on whether the child has inherited the affected allele or not. What if there are 2 affected parents? then you can tell that, there are 3 ways that a child could be affected. They could have 2 affected alleles and either be, it could be very severe when it's, or it could be the same as just having 1 allele. So this combination of having 2 affected alleles is called strictly dominant. Or they could have a combination of 1 affected allele and 1 unaffected allele, and in which case they could still be affected. They'll still be affected, but may or may not be as severe as the combination of 2 affected alleles. And then there is a 1 in a 4 chance of them not being affected at all. So take some time, look at these things, and I know it's new information and could be quite a lot of information, but take some time to absorb this. And if you've got questions, we can always talk about that in a bit more detail later on. Now, there are also a few special conditions in dominant inheritance called semi-dominant and co-dominant. Semi-dominant or incompletely dominant is when homozygotes, so identical in both copies, or compound heterozygotes, where both maternal and paternal copy are affected, but they have different variants. When both the copies, the maternal and the paternal copy, are affected, they're more severely affected than the ones who carry just 1 copy. So we talked about that in the Punnett table before. So if they carry 2 affected copies, they could be more severely affected compared to people who carry only 1 affected copy. So that is called semi-dominant or incompletely dominant. Pure dominant is when homozygotes or compound heterozygotes show the same phenotype as heterozygotes. So it doesn't matter if you carry 2 affected copies or if you only carry 1 affected copy, you have the same phenotype, and this is very rare. And co-dominant is when both alleles, paternal and maternal, together determine the phenotype of an individual. So for instance, depending on whether you've got OO genotype, AA genotype, AO genotype, BB genotype, all of those combinations give you a different phenotype of your blood group. So blood group is a co-dominant, so they both are necessary to determine the phenotype. So both the maternal and the paternal allele in different ways, but it's a different type of dominance. So it's a trait that occurs depending on what you've inherited from mother and the father, but it's not necessarily a recessive condition. Let's talk about X-linked inheritance. There are very few Y-linked genes that are of any relevance, so other than sex determination. So we won't really be discussing Y-linked inheritance here. It's not necessarily that relevant. There are some very rare genes, but it's not that important for us to know about that. But there are more than 300 diseases associated with problems in the X chromosome. So let's look at the Punnett table. It's not necessarily a Punnett table, sorry. This is just a table to show the different combinations, the genotype combinations and the phenotype combinations that you can get in X-linked inheritance. So the uppercase H denotes the wild type and the lowercase h is the disease allele. So you can see here that if a male is hemizygous, because they've only got 1 X chromosome, they're always hemizygous. If they're hemizygous for the wild type allele, they're unaffected. If they're hemizygous for the affected allele, then they're affected. So if the disease allele is present, they don't have another X chromosome to compensate for it, so they will be affected. In females, if they're homozygous, so both the alleles are unaffected, as in they don't have a disease allele, they have wild type allele in both the copies, then they're unaffected. If they've got 1 wild type allele and 1 disease allele, they're carriers, may or may not be affected, and we'll talk about these situations in the next few slides. But if they're homozygous, which means both the copies are affected, both the copies are disease alleles, then they're phenotypically also affected. Another unique situation with X chromosomes is that there is something called X inactivation. In females, although they have 2 X chromosomes, most genes in 1 of the X chromosomes will be inactivated. This inactivation may be different in different cells and different tissues. So if there is a disease-causing variant in 1 copy of the X chromosome, very likely that could be silenced. So that's why many carriers don't necessarily manifest, although sometimes there is skewed X inactivation. So SKEWED, skewed X inactivation, in which case carrier females may also be affected. So X-linked dominant and recessive, these are not seen too commonly in clinical practice. Most of us lump the two of them into X-linked, but it's also important to know the difference between X-linked dominant and recessive. X-linked dominant and recessive do not act the same way as autosomal dominant and recessive. X-linked dominant is when carrier females consistently manifest the phenotype, because even in females, even if they've got only one copy that is affected, they will manifest even if it is mild. X-linked recessive is when a phenotype is not observable in carrier females due to X inactivation of the affected gene. However, as we talked about random X inactivation or skewed X inactivation, sometimes females can have milder phenotype even in X-linked recessive. Therefore, it's not always possible to distinguish between X-linked dominant and X-linked recessive. So this is what X-linked recessive looks like. So as you can see, the dots are basically non-manifesting females. So they're carriers, they carry the genotype, but they don't have the phenotype. So they've inherited an affected genotype from their father, but they don't actually have the disease, but they can transmit it to their sons, whereas their daughters will be carriers. The sons will actually have the manifestation. The daughters, once again, if they have sons, they can actually have the manifestation. So one of the examples of an X-linked recessive condition is hemophilia A. So X-linked recessive inheritance, the incidence of the trait is much higher in males than in females. Heterozygous females are usually unaffected unless there is skewed X inactivation. Affected males' daughters will all be carriers. Any of his daughter's sons has a 50% chance of inheriting it. It's never transmitted directly from father to son because a father only gives his Y chromosome to the son. A significant proportion of isolated cases are due to new mutation. And as we talked about earlier, an example is hemophilia A. X-linked dominant. So this is an example of X-linked dominant pedigree where both males and females can manifest the disease. Females could be milder, but the difference between this and an autosomal dominant is that there is no male-to-male transmission. You see that the sons are unaffected of a male, but it's very difficult to differentiate between an X-linked dominant inheritance pattern and an autosomal dominant inheritance pattern. An example of an X-linked dominant condition is X-linked hypophosphatemic rickets. So we'll leave it there for now, and we'll talk about the rest of this topic in the next lecture. If you've got any questions, please email education@pansma.org, and I will try to answer your questions in a revision lecture as well.